morning, everyone. I have the pleasure today to present Professor Hagai and host, guest of Professor Sironi, to whom I leave his presentation. So have a good seminar, and thank you for being here.
Hi, everybody. Thank you so much for coming. So it's a pleasure to introduce Tzachi. Tzachi is a PI at Tel Aviv University. He did his PhD at the Weizmann Institute, and then he moved abroad to San Francisco and the Sanger Institute for postdocs. He has contributed very important papers in the field of virus evolution, protein evolution, and comparative transcriptomics. So it's a pleasure, and Tzachi, the stage is yours.
Thank you, Manuela. Thank you all for coming. Can you hear me? Okay, so this is the first time I'm trying this presentation. So feel free to interrupt with questions, comments, and things that don't look right to you. Let's start. So today I'm going to talk about some lessons we think we have about the evolution, the very interesting and bloody evolution of the interactions between us and our viruses. In general, we work on this very interesting topic. I think I'm very biased. Very interesting topics of host-virus coevolution. We work on two different aspects. Here to the right, we work at the cellular level. We're asking how different host species evolve to efficiently inhibit pathogens, and we know that different host species react completely differently to different pathogens, to the same pathogens. For example, as you probably know, coronaviruses, we think they originate in bats, and some bats are well adapted to coronaviruses and they do not become ill. Other species of bats, completely unrelated species of bats, are the natural reservoir of Marburg. Marburg is the even more virulent cousin of Ebola. We don't want to get it, but these bats, if they get it, nothing really happens. The virus replicates and somehow they manage to live happily ever after. So we are trying in that sense to understand how our immune system has evolved across different species to inhibit pathogens differently. And we try to capture the delta, the differences between the species, and to understand what is unique, for example, to the bats in their ability to inhibit specific viruses. In the other side of the lab and also of this slide, we're focusing at the protein level. We're asking how viruses have evolved to successfully replicate in the host cell. This is what I'm going to talk about today. So when a virus such as this guy here, which is HIV, gets into our cell, because it's an intracellular parasite, it's an obligate parasite that only can grow in our cells. Basically, once it starts replicating, it has to recruit many proteins, our own proteins, in order to hijack machineries that are needed for replication. And it also needs to dampen everything we put on its way to replicate, because we also have our own defense mechanisms within our cells that are trying to inhibit the virus from replicating. So at the end of the day, almost every viral protein forms numerous interactions with their host proteins. And the state of the art in the field is that for many of the viruses, although not for all of them, we have this interaction map, protein-protein interaction map. And I have here several examples from different viral species, where you have, for example, this is flu, you have the viral proteins at the center, and then you have many human proteins that were pulled down in this assay and are thought to interact with this viral protein. And again, they are forming these interactions, the viral proteins, presumably to hijack these various proteins, these human proteins in these cases, so that they can use them for their own replication. Now, what is nice or not so nice about these interactions is that they always lead to a genetic conflict. Why is that? Because most of these protein-protein interactions are beneficial for the virus. They are good for the virus, but if they are good for the virus, by definition, they are bad to us. You know why, right? You all got flu or cold recently or not so recently. So this is beneficial interaction for the virus. It is deleterious interaction for the host. However, some are deleterious for the virus and beneficial for the host. This is the specific case where you have a restriction factor, a human protein that tries to sabotage the virus replication in all sorts of various cool ways. The coolest human proteins, I think, and again, I'm very biased, are these restriction factors that are going and trying to attack viruses directly in all sorts of cool methods. But regardless of whether they are beneficial or deleterious for the virus or the host, they always lead to a genetic conflict because they are always beneficial for one side and they are always deleterious for the other side. So we have a conflict. Now, because we have this conflict, this very antagonistic interaction between the host and the virus, it is often perceived that the interaction side, the interfaces, the residues that come in touch between the host protein and the viral protein would evolve rapidly. And this is true in two different scenarios. Intracellular interactions, which is what I showed you before and what I'm going to talk about today mostly. So the interfaces between the host protein and the viral protein should evolve rapidly according to this notion. And also we have this concept of antigenic evolution where we have the antibody that tries to go for the viral capsid, and then the capsid is trying to evade it through mutations and evolution to get rid of the antibody. And this is well known, and there are all sorts of reviews and analysis about that. Now, why is that the case? Why do we think they evolve rapidly? So if we take the evolutionary time, which is this axis here, and we have two proteins. Let's say the viral protein wants to interact with the host protein in this case. So it's beneficial interaction for the virus, deleterious for the host. In this case, let's say this is the spike protein from corona. We all know it, we all like it. And this is ACE2, the receptor that the spike wants to interact with in order to enter our cell and start replicating. So of course, this is deleterious for the host. And if it's deleterious enough, if it exerts enough selective pressure on the host, then by random mutation and forces of selection, the host will mutate a way to lose this interaction, and then the host wins and the virus loses because the virus cannot, in this specific case, get into our cell, and it's great news for the host, bad news for the virus. This indeed happens in the course of evolution. We're talking here about millions of years, by the way. And then, two can play this mutational game, sadly. So interaction can actually be regained by the virus having its own counter mutation to have the interaction again. And the bottom line is that in the course of evolution, we will get this rapid evolution, rapid changes at the interface, mutations here and mutations here between these sites that are touching each other. And we call this situation an evolutionary arms race or Red Queen hypothesis. It's all the same. And this is all originating in the fact that they oppose each other. And we can often recognize them by looking at signatures of positive selection, which is a type of test that we can run in molecular evolution, where we're looking basically at a closely related species, in this case, human and primate, and find many non-synonymous mutations, many differences of residues in orthologous proteins, in the same protein in different species. So this is all nice and great. However, the problem in various studies, including by us, that showed that, in fact, when you take all the human proteome, you divide it into those human proteins that are known to interact with viruses and the human proteins that are not known to interact with viruses, those that interact with viruses are significantly more conserved than those human proteins that are not known to interact with viruses. So this seems to be exactly the opposite, in contrast to what I just told you about the great rapid evolution and evolutionary arms race and red team hypothesis. So how do we try to reconcile these contradictory results between highly conserved human proteins interacting with viruses and rapid evolution at interfaces between host and viral proteins? So our ongoing and previous hypothesis, we keep the same hypothesis all the time, that what we see here, we have contrasting trends that are a product of a mixture of interactions. So these mixture of interactions have different functional and biophysical and structural characteristics with different constraints. Not everything like humans. Some of us are better than others in some things. So some proteins can evolve rapidly and some proteins cannot evolve so rapidly. This is what we think. And we decided to take an approach because it's a very complex problem with many human proteins that interact with many different viral proteins. So we decided to try this approach of divide and conquer to focus on a particular type of host virus protein interactions that we can understand the biophysical nature of it and to understand how it is related to the constraint imposed on the interface. What I said now is probably not very clear, but hopefully it will become clearer in a few slides. And what we chose to focus on, which is quite weird, I think, was to look at interactions where the virus utilizes mimicry. And viruses, they're not nice or anything. When they want to interact with host proteins, they need a way to integrate into our interactome. And one way to do it, one simple way to do it, is to fool the human proteins into thinking they're interacting with the same human proteins they're familiar with by act of mimicry. So they basically mimic a protein or an interface of a protein in different ways. And our story today, I will divide it into two. We'll talk about, first of all, we'll talk about perfect mimicry, where viral proteins mimic short linear motifs that also exist in our protein. And in the second part of the talk, I will talk about imperfect mimicry of full host domains, again, by viruses. Let's start with perfect mimicry of motifs. So our proteins can interact with each other, and they love to interact with each other in all sorts of ways. One way that was discovered about, I would say, 20, slightly more than 20 years ago, is by domain motif interaction. So as you probably all know, we have in our human proteins disordered regions, these tails that look like spaghetti, boiled spaghetti, right? I need to be careful with this type of analogy in Italy. I think this is how it looks like, I think. Yes. So within these disordered regions, we have these islands of order that are motifs. These are repetitive motifs that can exist in many different human proteins, and they can interact specifically with another domain. So basically, these motifs are embedded in disordered regions, like this guy here in P, and it fits perfectly a binding pocket within a much larger folded domain. And these motifs are interacting with specific domains. So there is a matching type here. And you actually probably all are familiar with them. So SH3 domain, SH2 domain, TBV, WW, and many others are actually these peptide recognition domains, or in short, PRD, and they interact with specific motifs. This is the real motif. So if you have a protein where in its disordered region, you have a residue that is either an R or K or Y, then something, something, then P, then something, something, and another P. Yeah, that is as simple as that. This guy, given the right condition will specifically interact with the binding pocket of an SH3 domain. And that, as unimpressive as it may look, is actually a very common way to interact between our proteins. And this is very important for regulatory interaction. So many proteins you're familiar with, many pathways in cancer, are actually mediating their interaction through this type of domain motif interaction. And sadly for us, this is also a great way for the viruses to integrate into our interactome, because these motifs are small, very simple, you saw them. So they can easily emerge what we call de novo in the viral protein, because it's very easy to mimic these 2 or 3 or 4 residues, and it doesn't take much space in the viral genome. And so a virus can come up with these motifs that look exactly like our own motifs and specifically interact with a human domain to hijack this pathway, this cellular pathway for whatever it needs, the virus needs. And sadly for us, it is very common across many viral proteins and many different viruses, this motif mimicry. This is a real example, this curly guy here called retinoblastoma domain, which is associated, of course, with the retinoblastoma disease. And you can see here that into its binding pocket perfectly fits a viral motif, only 3 letters, from simian virus 40. But it looks exactly like our own motif. So the simian virus 40 protein manages basically to hijack this pathway very easily, very cheaply. And the really good thing about this type of interaction is that we don't need to have very difficult or challenging tools to use to predict that this interaction happens, because we can easily identify the motifs in the viral protein, and we can easily identify the matching domains in the human protein. So we can very easily say, if you tell me, okay, you have here a pair of human protein and a viral protein that interact with each other, and then I say, okay, fine, let's look for motifs in the viral protein and for matching domain in the human protein. If we find a match, we will say bingo. We know, we presumably know how they can interact with each other, which is the only interaction today, even in these fancy days of AlphaFold, that we can predict well the interaction and the interface, the exact residues that interact with each other between human and viral proteins. Trust me, we tried that. So what we did with that, going back to our question about the evolution of interfaces, we took the whole human proteome, 20,000 genes or so. We split them based on whether they have these PRDs, these peptide recognition domain, at least one domain within the protein, like the SH2, SH3, PDZ, and all the rest, which we call non-PRD, so PRD, non-PRD, about 10% are PRD. Then we overlaid it with publicly available data of human virus protein-protein interaction. From the literature, there are thousands of studies of protein-protein interaction. You see, it's about 1,300 of these PRDs, these human proteins that have these domains that bind motifs, are known to interact with at least one viral protein, viral binding PRD. But we don't know how. We just know that they interact. And then we did this bingo matching, as we said. We're looking at the human PRD to see which domain it is. We're looking at the virus to see if there is a matching motif. So if there is a motif that knows how to bind an SH3 domain, and this human protein has an SH3 domain, we will say, bingo, we know, we infer that they bind each other through domain motif interaction. And from this set of about 300 proteins, human proteins, we have about 10% that are experimentally validated by hard-working virologists that did virology, biochemistry, and many other tests and studies to verify that indeed these viral proteins interact with the host protein. It's actually very challenging stuff to do. And we also have on the other side of the non-PRDs, the human proteins that don't have any of that domain, we have still non-PRDs that are known to bind virus. So we have these many subgroups of human proteins. And what we did, we looked at their rate of evolution across vertebrates, to be accurate, across amnions, but sort of the same. We did it using a measure called dn over ds. It doesn't really matter, but we have here several orthologs going from human all the way to chicken and Galapagos turtle. And we can use this measure to look at the evolutionary rate of all the proteins in a comparative manner and every residue in a comparative manner. And the higher it is, the faster the evolution across vertebrates. What you see here is that all the purple guys, which are the groups that bind viruses, are lower. These are box plots, so these are distributions. All the purple guys are lower in their median with respect to the whole human protein in gray here, which means that indeed, by and large, human proteins that bind viral proteins are more evolutionarily conserved. This is statistically significant, and it repeats what we said and others before. What we see when we are looking at the PRDs, the human proteins with these domains, is that they are significantly more conserved than non-PRDs. And if they bind viruses, they are even more conserved. And if they are inferred to bind viruses through domain-motif interaction with this bingo approach, they're even more conserved. And if they are experimentally validated to interact with these motifs as part of their application, they're even more conserved. So the bottom line of this analysis is that viruses evolve this motif to target super, extra, ultra conserved proteins within our interactome. Bad news for us, not the end of the bad news for us. So because it's a very nice system, we can actually look at the protein and deconvolute the human protein into the domains that bind these motifs, the PRD, and to the non-PRD domains. And we can also look at specific regions within the PRD. And what we saw, and I'm not going to show you any more graphs because it will take us too long, that almost always the domains that have these, that bind these motifs are more conserved than overall the protein that they are at with respect to other domains that are not binding. So the domains are more conserved than the protein, which is already pretty conserved. Now, we can also partition the domain, this PRD, into core residues, interface residues that are touching the motif and the surface. And what we saw that the interface, the stuff that touches the motif, the residues there are highly conserved. And I will say, first of all, surprisingly, they are as conserved as the core, which is quite funny because they're exposed. Usually this is not the case. But also, if you look at those binding pockets and compare between binding pockets that are known to interact with viral motifs and the same binding pockets that are not known to interact with viral motifs. For example, we have SH3 domains in many human proteins. Some are known to interact with viruses, some are not known to interact with viruses. We can compare them. So the interfaces of those that interact with viruses are significantly more conserved. And if you partition those that are known to interact with viruses into those that are known to interact with one single virus or with many viruses, those that will interact with many viruses are even more conserved. So many different viruses have convergently evolved to target extremely conserved binding pockets within these conserved domains, within these overall conserved human proteins. A lot of conservation. And we were asking ourselves why. So there are many reasons why these human proteins can be conserved. This is one probable reason. We here counted the number of human-human protein interaction. So we take every human protein and we count how many proteins it is known to physically interact with. So this is from the string database. We assemble everything that is there. And what you see here, this is the overall human protein on average. A human protein is known to bind to 200 proteins. And you see that you look at the PRDs, the ones with the domains, and the one that especially bind viruses goes up 3 times. So the human proteins that interact with motifs are hubs. They interact with many other human proteins. And we can do the same bingo analysis on them to look at this protein-protein interaction and to say which one of them is known to interact through domain motif interaction. And almost all of what we see here are domain motif interaction, meaning that the same domain is used by hundreds of human proteins through the same mechanism, through this motif for some sort of interaction that is needed for the cell. And the virus comes with the exact same motif to interact with the exact same hub in exactly the same way. So obviously these things cannot really rapidly evolve because we are stuck in evolution, right? And this is basically a mirroring picture of the evolutionary rate that we just saw before. Now you can ask me, okay, so we are kind of so screwed. We cannot evolve very rapidly with this interaction. What happens from the viral end very rapidly? And for that, more recently, we asked the question, we took viral motifs from human infecting viruses, and we defined how conserved they are based on whether or not they occur in orthologous genes in viruses that infect other primates. This is all primates, but we're talking about the viruses that infect it. And this is a motif that is highly conserved. And here we have a motif that is conserved less. This doesn't exist in the last primate infecting virus. And this is even less conserved. It only exists in human and chimpanzee infecting viruses. And what we saw when we compared functional motifs to non-functional motifs, that the functional motifs are almost always completely present in all the human and primate infecting viruses. And this is significantly more conserved than non-functional motifs controls that we're looking at. This is the case of cytomegalovirus clade, and it's also true for other human infecting herpes viruses to the left and to the right. So basically the viruses also, once they evolve this motif that is beneficial for them, they don't easily evolve away from that, and they kind of also stuck with the motif, although, you know, being stuck with the motif doesn't pose such a constraint like on the host, where we are stuck with this harmful binding of the motif by the virus. So the bottom line of what I just said was that in the case of perfect mimicry of motifs, both the viral motifs and their host target domain are highly conserved across evolution. There is no such thing as rapid evolution, evolutionary armfree. Now, I mentioned that we are starting to also look at imperfect mimicry of host domains. So this is an entirely different system. We're talking about much larger mimicry of whole chunks of domains. This is the proper protein that you have in mind when you think about proteins. It's globular domains that can fold properly. And what do they do? Yeah. Now, these domains, viruses also can steal from us. They don't evolve them de novo like motifs. They usually hijack them through horizontal gene transfer to the RNA or the DNA of the host to the incorporated into the genome of the virus. And what is interesting, so going basically from this domain here, which we call host mimic, to the viral mimicking here. And basically what's very interesting, and we don't know really why, following viral acquisition, they almost always diverge very rapidly in sequence. So they maintain overall structural similarity, but the sequences are very rapidly going very, very low. And we think, but we don't really know, that they carry out the exact same function like the original domain of the human protein, the host protein. And some of them can be used for interaction with other human proteins, right? Exactly like the motifs that bind domains. We have these domains that can bind other domains through domain-domain interaction. And basically, we have this triangle of not much love, where we have a host mimic protein domain that basically interacts with this human domain. And we have this viral mimic that also interacts with the same host target. So we have a love-hate relationship here. And the question here, the big question here, how do they evolve? Because you have the same exact problem like before. If the blue guy will try to evolve away from the red guy, the viral mimic, that will be great because we avoid the virus. But it will be really bad because then we can also not interact with the purple guys, the host mimic. So we have a problem. So what's going on? And in order to answer this, this is not as simple and as nice as the domain motif interaction. We use AlphaFold. We took viral proteins, all viral proteins from 5 different human infected viruses. We ran AlphaFold to predict their structure. We then ran FoldSeq, which is the structural homology tool, to find basically the purple and the red protein. So the red is the viral mimic, and it's a structural homologue of the purple human protein. And we also can, these are the statistics, how many pairs of proteins of viruses we have that behave like mimics. In this case of these human viruses, with a substantial number of proteins that mimic human proteins or host proteins. Now, we also have a missing link here. We don't know which of them interact with the same target like their structural homologue human protein. In order to figure out that, we took publicly available data of both within human protein-protein interaction and host virus protein-protein interaction, looking for these mutual targets between them. Essentially, if we have a protein that is known to interact with both the host mimic and the viral mimic, we call it a mutual target. Okay, so this is all based on publicly available data from the literature. And yes, it's quite noisy and problematic. But still, we have these triads of human mimic, human target, and viral mimicking proteins. And we run on all of them evolutionary analysis to, again, to compute the evolutionary rates at the level of residues and at the level of the whole gene. And then we can ask evolutionary questions about the evolutionary rates of these different proteins. What you have here, this is across primates evolutionary analysis. And again, higher means factor. This is the whole human proteome. What we can see here is again that the viral binding human proteins are significantly more conserved than the rest of the human protein. The target, these guys in blue here, are significantly and also in absolute value lower in their evolutionary rate than all human proteins, as well as the subset of viral binding human proteins. So the targets, these mutual targets in blue, constitute a highly conserved group of human proteins. The mimic guys in purple here are actually significantly, but only slightly in terms of absolute level, less conserved than the whole human proteome. So the targets, the blue targets, which is the bottom line here, are again highly conserved. What we did, we took these guys and we looked at how many of them show signatures of positive selection, meaning they have some evolutionary arms race, there is some, presumably evolutionary arms race, they have some positively selected residues within them. What you can see here, that the viral binding group is actually significantly enriched with positive selection. So we have slightly higher fraction of positively selected human proteins in the viral binding that show signatures of positive selection with respect to all proteins. The targets, the blue guys, are again, they're significantly depleted of signatures of positive selection. This is in contrast to the superset of viral binding, because targets are also viral binding, right? And the mimic again show weird results. They're slightly higher than all proteins, but this is not significant. So the bottom line in here is that the target proteins, the blue proteins, they were told not to move. The blue proteins are highly conserved. They have lower DNDS values, and they are significantly depleted in the fraction of positive selection with respect to all human proteins and also with respect to the viral binding proteins in general. Now, because we can solve the structure of some of these complexes between the mimic and the target, or between the mimicking viral protein and the target, we can look at different regions of the interfaces between them. We can, for example, say this part is the interface that involves the mimicry interaction, and this part we can predict it's an interface region in general, meaning it's not, it is an interface region, but not for the mimicking or the mimicked protein, using some program. And what you see here is that what we call the general interface or the control interface are always more rapidly evolving than the interfaces that are involved in the mimicry, both in the target in blue and in the mimic in purple. So basically, these interfaces that are involved in mimicry are more highly conserved than other interfaces on the surface of the same protein. I think it's nice. We also can take this triangle and ask, okay, so we have the mimic and the viral mimic. They are both interacting to some extent with the same interface, but there is difference. Remember, I said they are never exactly the same. This is imperfect mimicry for whatever reason which we don't fully understand, which means that this figure is wrong. We basically have some residues that only the mimic interacts with, and some residues that only the mimicking viral protein interacts with. So we can deconvolute this and say, let's take the mutual region of the interface separately from the viral specific and separately from the host specific interacting residues. And what we see here, it is not as statistically impressive as the previous slide, but still, what you see here is that the mutual interface is the lowest. It is the most highly conserved or constrained region in the interface, meaning the region that both the host and the virus interact with, or if you want, the region where the mimicry is perfect, is the most highly conserved. Summary. So we talked today about two different mimicry systems. I apologize for this bombardment of colors. We had the motif mimicry in the beginning, then we had the domain mimicry at the end, perfect mimicry, imperfect mimicry, small de novo evolution of mimicry, large horizontal gene transfer in the case of domain mimicry. However, in both cases, we saw that the motif mimics and the domain mimics of the viruses target highly conserved host targets. That is true for both of them. In addition, these are binding to regions within the targets that are highly conserved. We saw it at the case of the PRDs, at the case of the domains that bind motifs. We saw that the binding pockets are extremely conserved. They actually are frozen in time. If you compare residues in this interface between human and Galapagos turtle, you will find no difference at all, nothing, zero. This is almost an evolutionary frozen in time interface. And this is also true, apparently, for the domain mimic, where we saw that the parts of the interface that are involved in the mimicry are more conserved than what we call general interfaces. And also, when we look at the mimicked and mimicking in very specific interactions with residues, we saw that the neutral region where they both interacted is the highest conserved, it's the most conserved region. What we don't see almost never is evidence for positive selection. So if anything, there is depletion of positive selection in target, and there is an unclear trend in the case of the mimicked proteins here in the purple. This is also true. I didn't talk about it, but this is also true here. In the case of the PRDs, the domains that bind motifs. Of course, there are some post-viral interactions that involve positive selection. When we look at them specifically, we see completely different things. They usually do not involve mimicry of the viruses. If they do involve mimicry, proteins, they don't usually interact with the parts that are the mimics. They interact with other parts that are not the mimics. Those human proteins that evolve rapidly, that have signatures of positive selection and interact with viruses, usually interact with very few other human proteins. And the specific residues that evolve rapidly, that have these signatures of positive selection, are free to roam and to change. We see that in the structure, they form very few contacts with other residues within the structure, which means it is easier for them to change from one residue to the other. So everything I'm showing here is basically telling you one thing, that if you're under constraint, meaning if the virus is taking advantage of a similarity of interface, you cannot evolve away because you're stuck. You need to do your daily job, whatever it is. You need to interact with host protein in the same manner. If the virus takes advantage of that, you as a host will need to come up with a better solution. You cannot evolve away. The only cases where we have signatures of positive selection are these few cases where human proteins can, or the host protein in general, can evolve the interface without jeopardizing the rest of the interactome. So in general, if we should think about the evolutionary trends of host-virus interactions, perhaps we should rethink it. Maybe instead of thinking about Alice and the red queen hypothesis, we should move onward to a newer Disney movie, newer Disney princess, and think about evolutionary frozen in time interfaces, or the situation that we call evolutionary stasis. And I just want to thank everybody in the team that were involved in this work, and our collaborators in Italy and across the globe. We also have here people from Canada and from Germany. And thank you all for coming and listening. That's it. Did I manage to do it?
Some questions? I will do the microphone. Okay. Sì, per pochino, ma vai.
So thank you very much for your presentation, very interesting. So I was wondering, I have a couple of questions actually. I was very curious to know whether, so you showed us that all your analysis is based on predictions, right? Right. So I wonder whether, how can you check that, no, you can do a validation on these type of predictions in order to really understand whether these interactions are real and not just a model, you know. So that's the first question. And the second one is, how can we use this information, which is a huge information, if these interactions are real? Because, of course, it's interesting to know from an evolutionary point of view, of course, but because I'm an immunologist, so I'm on the host side. Okay.
I'm also on the host.
Okay. And therefore, we always like to see how we can use this basic information to translate in something that then we can hijack. so that we can combat better the viruses?
Good question. So first of all, regarding experimental validation, we are trying, cannot always do that, but we're trying to also look at experimentally validated cases where we even have structures or that were used in a completely different way. For example, in the case of motifs, they're almost following the submission of this paper, but before it was accepted, there was a paper that basically looked at corona interactions from disordered region, and basically they predicted, based on their own experiments, data of motifs, of viruses that interact with the host, and we use that data to show that we see the same results. So these guys are clever and they evolved to target highly conserved regions. And we also see it when we're trying to use data sets that are basically only based on mechanistic studies with, very, very meticulous point mutations in the viral genome, in the host protein, and then doing biochemistry and viral replication. This is why we only have a few dozens of them. They are very difficult and challenging to do. And we see the same trends. So I don't think we have any bias in terms of our analysis, at least in the sense of the motifs. In the case of the domains, things are much more challenging. We have extremely few, this is annoying, but that's the situation. Very few complexes that are solved, experimentally solved, and we rely on all sorts of assumptions or on AlphaFold multimer, which is the version that can give you a complex, which is the best in the market and even got a Nobel Prize, but it's not very good in host virus. We actually have a very small fraction of complexes. This is an open question. I can also say that we have a problem of exploring. One question can be, are these interactions really important for the virus? The problem is we know from many viruses that you can take off a whole protein from them and they would still replicate in vitro. That doesn't mean that they will replicate in vivo, because usually they will get rid of the protein if they don't need it in their gene. So we have a real experimental problem in studying the importance of all these interactions in vivo. Now, what can we do with that for humanity? So our, ideally, what we can see here, we can find highly conserved interaction modes between different viruses and human proteins, which can be a basis for universal antiviral treatment to block these interactions. So this is a very different approach, and blockers of protein-protein interactions between hosts and viruses have not been the focus of the antiviral treatments, not only immunology, but like if you think about how we inhibit HIV, that is not how we do that. So that is something that maybe it's time to start pushing, and I know some people are thinking about that. The other thing here, what we show is that actually evolvability is not something that happens a lot, despite our perception about it. And what we are trying to understand is where this evolvability happens. And I think if we manage to expand it to enough systems, these are just two, maybe we can even predict how viruses can evolve and emerge. Then we can even tell maybe what would be the next coronavirus. Maybe.
Tzachi, thank you for your very nice presentation. I'm a pharmacologist, so while you were talking, I was thinking how we can prevent viruses from replicating. And at first, I was thinking about this highly conserved region that we can target. And then while you were talking, I was thinking, no, it cannot be, because those are exactly what we use. So I was wondering if your approach can be used to look for less conserved region, for something which is typical of a virus, not of a human counterpart. Do you think that we can use something like that in order to find new targets?
So I think that we can definitely do that. We have a bigger question about modes of evolution, of slow evolution in the host, slow evolution in the virus, rapid evolution in the host, rapid evolution in the virus. Does it have to be symmetrical? So you have all four scenarios. This is we are still working on it, but definitely some of these scenarios should go in line with your idea. But I warn you and myself and all humanity that we are not as clever as virus evolution, and we can often find a way around, like in Jurassic Park. So we can try. I do think that if we find enough blocking steps and treat simultaneously with several drugs, then maybe we can block effectively the viruses.
More question?
I have a maybe naive question. If most of the binding surfaces are conserved between virus and host, does this mean that viruses can easily spill from one species to another?
Is a great question that it's not naive at all, and I have no good answer to it. We're trying to understand that. We're actually now looking at a data set that was, it's amazing how difficult it is to study, because the data is not organized in any way that you can actually look at it. There was a recent paper by Francois Ballou, who created these trees, evolutionary trees of viruses with zoonotic jumps identified, and we're hoping to exactly look at exactly what we said. Okay, so there is a zoonotic jump. What are the differences with respect to before and after, and with respect to the whole tree? I don't know. I really don't know. And also keep in mind, we're talking about viruses. Different viruses are more different than we and Drosophila are. So we need to respect them, and each of them can have its own way of jumping.
More questions? No? So thank you for all. Very interesting. Terrific. Thank you. Oh, sorry. Just one, another naive question.
Yes.
For you about another kind of mimicry, which has the immune response that